Avoid these statistical mistakes in oncology clinical trials. Top 10 statistical mistakes that can be seen in oncology clinical trials. Number 1. Inappropriate endpoint selection. Choosing endpoints that don't reflect clinical benefit. For example, relying only on tumor shrinkage instead of survival. So here the tumor shrinkage and survival both are important, not a single endpoint, right? So you need to align these endpoints OS, PFS and ORR with the study objectives. That's how you need to fix this mistake. Now say for example, I have a lung cancer trial that shows high tumor shrinkage, right, ORR, but patients don't live longer. Shrinkage is there, the tumor has reduced, but the overall survival is unchanged. That means the survival of that specific patients is not increasing even though the tumor has shrinkage. So the mistake here is the endpoint does not truly reflect the patient benefit. So the lesson here is you need to choose the endpoints that matter clinically. OS, PFS and ORR. All these endpoints which are there, which are critical in oncology, they need to align with the study objectives, and the survival also should be seen, the tumor shrinkage also needs to be seen, and there has to be the specific parameters that needs to meet, so that you can see the patients are getting benefited by consuming this oncology molecule. Number 2, underpowered study design. Now, small sample size leading to inconclusive results. Now, these are very common in rare cancers, but they are still risky. So, you can fix this by performing proper sample size calculation with realistic assumptions. Say, for example, a rare cancer trial enrolls only 40 patients. Treatment effect looks promising but not statistically significant. So the mistake here is sample size is too small to detect the real effect. So the sample size needs to be sufficient so that you can get promising results, as well as the you can able to see the significance statistically, right? So the lesson here is to ensure that this sufficient power is determined and the justify limitations clearly, right? So sample size needs to be appropriate so that you get the real effect. Number 3, ignoring multiplicity adjustment. Testing multiple endpoints without controlling type 1 error. Now this can lead to false positive conclusions. So you can fix this by using methods like Bonferroni, Holm or hierarchical testing strategies. Now say for example, there are trial test OS, PFS, ORR and other 5 biomarkers all show significance, right? But the mistake here is there is no adjustment, so which leads to false positives likely. So the lesson here is you can control type 1 error by using the hierarchical testing. Number 4, improper handling of missing data. Now, when you ignore dropouts or censoring patterns, like say 0 and 1, which are the censoring and the uncensoring values that you assign to the specific patients or to the records. So when you ignore this, what happens is, especially with these are critical in survival analysis. So you can fix this by applying censoring rules correctly and consider sensitivity analysis. Now say for example, there are many patients who drop out due to toxicity and the analysis ignores them. So in statistical analysis, you are not including the dropouts and you're not including the censoring patterns, which is 0 and 1, which is required when you do the survival analysis. So the mistake here is you are doing the biased survival estimates. That means you are including only censoring values and you're not including the uncensoring values. So you are doing the bias of this data. So the lesson here is you need to apply proper censoring in the sensitivity analysis. Number 5, misuse of survival analysis methods. Incorrect application of Kaplan-Meier or Cox models, which generally violating proportional hazards assumption. So we need to fix this by checking assumptions and consider alternative models for example stratified Cox or RMST. So say for example the Kaplan-Meier curves crosses. Usually the Kaplan-Meier curves, they don't cross. You will be able to see these stepwise curves with, they are either increasing or they are decreasing, right? So that's how they are not supposed to cross each other, especially the Kaplan-Meier curves. But the Cox model still used blindly. So the mistake here is you have violated proportional hazards assumption, and the lesson here is to check assumptions or use alternative methods. The Kaplan-Meier curves, it's very important, and looking at the curve itself, anybody can come to know whether the treatment is effective and are there any survival chances of the cancer related patients. It can be easily seen in those curves. So that's how the graph should be in oncology. Number 6, overinterpretation of subgroup analysis. Now, drawing strong conclusions from small subgroups, which leads to high risk of false discoveries. So, you can fix this by treating subgroup findings as exploratory unless pre-specified. Now, say for example, the drug works only in a small group. Say for example, 15 patients claimed as only the target population. The mistake here is you are doing the random chance mistaken as a real effect, right? So the lesson here is you need to treat subgroup findings as exploratory, right? So you need to ensure that you don't over interpret the results by using only small subgroup of the patients. Number 7 is lack of randomization or poor stratification. Now, there is an imbalance in baseline characteristics. So, bias in treatment effect estimation. So, you can fix this by using proper randomization and stratify by key prognostic factors, say for example stage or biomarker status. So say for example the treatment arm has more advanced stage patients than controlled, right? So the mistake here is there is an imbalanced bias results, and the lesson here is to stratify by key factors, for example stage or the biomarkers. Number 8 is failure to account for competing risk, ignoring non-cancer deaths in survival analysis. Now, any patient that has died during the clinical trial, whichever the therapeutic area it is, the first important is within 24 hours that death should be reported to the regulatory agencies and to the concerned departments, especially the sponsor, if any death has occurred. So when you ignore these, so that happens to creating the risk, and that leads to biased estimates. So you can fix this by using competing risk models, for example fine gray model. Now say for example elderly cancer patients die from heart disease, but not by consuming the oncology clinical trial molecule which is under testing, right? But analysis treats them as standard censored cases. So the mistake here is you are overestimating cancer survival, and the lesson here is you need to use competing risk models. So you should be very ensure whether any death happens, and the censoring and uncensoring to any patient, it's very important in oncology clinical trials, because one value, if you change from uncensored to censored, for example for one record, your entire analysis can go wrong. So you need to be very precise, and very attention to detail is needed. Number 9 is inadequate interim analysis planning. Ad hoc interim looks increasing false positives, which is ethical and regulatory concerns. Now you can fix this by predefined interim analysis using alpha spending functions. So say for example, sponsor checks data multiple times and stops early when the p-value is less than 0.05. The mistake here is an inflated false positive rate. So you need to make here the lesson as predefined interim analysis with alpha control. Number 10, poorly defined statistical analysis plan. Now this leads to ambiguity in methods, endpoints or populations. Ambiguity is nothing but the uncertainty. There is no direction as to what needs to be done, because the definition of these methods and points, they are not defined properly in SAP, right? So that leads to inconsistent or biased results, because the programmer and biostatistician does not know like how to calculate that endpoint, or how to use method, what assumption needs to be done. So if your SAP is poorly defined, and the assumptions and the formulas and everything is not taken properly, then obviously it will lead to errors. So you can fix this by developing a detailed pre-specified SAP before any database lock. Now the statistical analysis plan, it needs to be finalized and approved with sponsor before database lock. Now database lock cannot happen unless and until the SAP is finalized, unless and until the SAP is approved, unless and until the SAP is digitally signed with sponsor and all the stakeholders. I hope this is a common needed requirement for the database lock, right? So say for example, after seeing data, team changes the primary endpoint from OS to PFS, right? At the last minute, you change the main primary objective. So the mistake here is you are doing this by data driven bias. Looking at the data, like the data is changing, so you change the endpoint at the last minute. So the lesson here is you need to finalize SAP before database lock. Database lock cannot happen unless and until the SAP is finalized, approved by sponsor and all the stakeholders, and until and unless all the stakeholders have given their signatures on the statistical analysis plan. That is the basic fundamental foundation. In oncology, time to event data plus complex endpoints plus regulatory scrutiny makes statistical rigor critical. Even very small mistakes can lead to trial failure, regulatory rejection according to US FDA or other multi-country specific agencies, and it can also mislead clinical decisions. And now all these things, it always impacts the human patients that are involved, and these mistakes can even cost the billion of dollars. Like you're investing dollars, and you're investing so much of money in one clinical trial to get that molecule launched in the market. So that very small mistake can give a loss to the huge amount of money, especially like taking the time, money, and the efforts by the various stakeholders. So you cannot decide properly, so it can lead to misleading decisions. Now you can imagine a phase 3 oncology trial where PFS shows significance, right? The overall survival does not. The multiple subgroup analysis shows mixed results. So nothing is consistent. All these three parameters, which are especially important in oncology, all three are giving you results in three different directions. So there is no like a common direction or a common results that you can interpret, you can write in your statistical analysis plan report, right? So all this is giving you three different direction results. So without proper multiplicity control and pre-specification, the study may fail regulatory approval despite promising signals, right? Many oncology trial failures are not due to bad drugs, but due to poor statistical planning and execution. Now, whenever you work any organization or any institute, like any government institute or any corporate company, being as a SAS programmer, statistical programmer, or a biostatistician, you carry a huge responsibility and accountability as to ensure that whenever you do any programming or any statistical analysis, that needs to be correct as per the in the going into the correct direction, so that you get the correct results. Because one single mistake, especially in the oncology clinical trials, it can lead to failure of that entire trial. Now that can bring the loss of the billions of dollars that are invested for conducting that trial, then all the stakeholders time and the efforts which are being done. And no matter, even if you have done your job properly as a SAS programmer, statistical programmer, and a biostatistician, but the other department has not done that job, job. So even that could also fail. It's not like only SAS, but since this topic is related to statistical mistakes, I would like to focus on this specific point. So being SAS, statistical and biostatistician, it's a responsibility and accountability when you're handling the human patient's life data, and you carry a kind of a responsibility that can either create the good benefit for the society when that molecule launches into the market, or like you would like to see the other way. So whenever you work on oncology clinical trials, and whenever you read any documents, be it SAP or any other interpretations, or whenever you do the review of the data, check thrice and ensure that it is going into the correct direction. No matter, even if you feel that the direction is not going into the right track, you see a major risk in especially the data, or like you're not getting the sufficient data, you can always raise as a concern to the your project manager or to your immediate team lead, so that can fix the issues and the problem at the right time instead of after the database log. Many thanks for watching my video, and happy learning. Please like, share, subscribe, leave a feedback comment. Many thanks for your support and having great connection.